What's up, biology students? Mr. Holloway here. In today's video, we're going to talk about another kind of organic molecule upon which all life on Earth depends. Today, we're going to talk about nucleic acids, the molecules that make up the universal genetic code of life. Like other organic molecules, nucleic acids are made up largely of carbon atoms. Carbon is a special kind of atom because it can form up to 4 covalent bonds with up to 4 additional atoms simultaneously. This ability allows carbon to form very large, very complex molecules, and it is this complexity that allows carbon-based molecules to do so many important jobs for and inside of living organisms. In addition to being composed of carbon atoms, nucleic acids are also composed of hydrogen, oxygen, nitrogen, and phosphorus atoms. The function of nucleic acids is to store and transmit genetic information. This genetic information is said to be hereditary because it can be passed from one generation of living organisms to the next, meaning that this information can be inherited by children from their parents. This is why children look like their parents, and why we can inherit certain traits, both good and bad, from our ancestors. That might mean that we inherit our mother's blue eyes or our father's curly hair, but it could also mean that we inherit our grandmother's predisposition for heart disease or our grandfather's thyroid disorder. Understanding the structure and function of nucleic acids can not only help us to understand why we look and act the way we do, it can help us to better understand how traits are passed from one generation to the next and may one day lead to cures for diseases and disorders that are coded for in our genes and which are currently incurable. In fact, genetics is one of the fastest growing fields of science, and we are making incredible discoveries about how nucleic acids affect our lives almost every day. Just like carbohydrates, nucleic acids are composed of numerous monomers linked together to form a complex polymer. The monomers of a nucleic acid are molecules called nucleotides, and all nucleotides are fundamentally similar. Every nucleotide is composed of a nitrogenous base, a 5-carbon sugar, and a phosphate group, arranged as shown in this picture. These nucleotides bind together with other nucleotides to form long chains, polymers that we call nucleic acids. There are 2 important kinds of nucleic acids, deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. As was the case with carbohydrates, monomers join together to form polymers through a process that we call polymerization. But the structure, and therefore the function of nucleic acids, is quite different from that of carbohydrates. Here we can see a DNA molecule, which is one variety of nucleic acid. DNA is actually 2 long strands of nucleotides, each running in opposite directions, and coiled around one another in a shape that we call a double helix. Here, where the double helix has been uncoiled, we can see the individual nucleotides that make up each strand of DNA. We can see that the phosphate group on one nucleotide bonds to the sugar portion of a different nucleotide, forming an alternating pattern of sugars and phosphates that we call the sugar-phosphate backbone. Nitrogenous bases stick out from this backbone, and we can see that the 2 strands are held together in the middle because of hydrogen bonds formed between the pairs of nitrogenous bases. You might remember that water molecules stick to one another and to other substances because of their ability to form hydrogen bonds. And these same kinds of hydrogen bonds are what holds the 2 strands of DNA together in a DNA molecule. The overall shape resembles that of a ladder, in which pairs of nitrogenous bases make up the rungs of the ladder, while the sugar-phosphate backbone makes up the side structures to which these rungs attach. Imagine taking that ladder and twisting it into a coil, and you'll have a pretty good mental image of what a DNA molecule looks like. All nucleotides are pretty much the same, except for the part that we call the nitrogenous base. And as it turns out, this is a really important piece. There are 5 different nitrogenous bases that a nucleotide could have, 4 of which are found in DNA, and 4 of which are found in RNA. That's one of the major differences between DNA and RNA. DNA nucleotides have a nitrogenous base made of either guanine, cytosine, adenine, or thymine, whereas RNA nucleotides have either guanine, cytosine, adenine, or uracil. These nitrogenous bases form hydrogen bonds with other nitrogenous bases, and this is what holds the 2 strands of DNA together in a double helix. But there are some certain rules that determine how this happens. For example, guanine, or G bases, can only form hydrogen bonds with cytosine, or C bases. Likewise, adenine, or A bases, can only form hydrogen bonds with thymine, or T bases. These are called base pairs, and we can see those base pairing rules illustrated for us here in this picture. This picture also shows us why these base pairing rules exist. It has to do with the fact that certain bases have complementary shapes, meaning these shapes fit together like puzzle pieces, and just like puzzle pieces, two bases with shapes that are not complementary will not fit together. Every living organism has DNA made up of the same kinds of nucleotides and the same 4 nitrogenous bases. Think of this as the 4-letter alphabet that can be used to write out instructions necessary for building a living organism. Just like we can put together the letters in our alphabet to form many different words and sentences, and use these to tell a nearly infinite number of stories. This is why we call it the universal genetic code, because all living organisms use the exact same code, made up of the exact same molecules. The only difference is the order and the number of nucleotides contained in an organism's DNA. Just like the difference between Harry Potter and the Hunger Games is due to the different order of words used to tell each story. As we will learn later in the year, this makes it possible to take DNA out of one organism and put it into another organism in a process that we call genetic engineering. And it is possible only because all living organisms use the same genetic code made of these same basic molecules. Pretty cool, right? We started our existence as a single cell. And this single cell, called a zygote, contained all the DNA necessary to build a complete living organism. As that cell divides to form more cells, the DNA inside is able to replicate itself so that each new cell gets a full and complete copy of the DNA found in that original cell. As we can see in this picture, by unzipping a molecule of DNA into 2 individual strands, new bases can be paired up with the old bases to form 2 new DNA molecules, each identical to the original because of the base pairing rules we just talked about. Each one of these new DNA molecules is made of 1 original strand and 1 new strand. The ability of DNA to precisely replicate itself is what allows genetic information to be passed from one generation to the next, in much the same way that DNA is passed from one parent cell to each of the daughter cells produced during cell division. When a cell divides, and DNA is passed from the original parent cell to each of the 2 new daughter cells, the DNA that is passed on is completely identical to the original set. We call this form of reproduction asexual reproduction. During sexual reproduction, however, DNA from one organism is combined with the DNA of another organism to form a completely new combination of DNA. To accomplish this, organisms that reproduce sexually use special cells called gametes. Male gametes are called sperm, and female gametes are called eggs. Unlike all of our other cells, which contain a full and complete set of DNA, gametes contain only a half set of DNA. This way, when a sperm cell that contains a half set of DNA combines with an egg cell that also contains a half set of DNA, a process we call fertilization, the resulting cell, called a zygote, contains a full and complete set of DNA. Half the DNA inside this zygote came from the female parent, and the other half came from the male parent. This is why we look a little bit like each one of our parents, but also look unique in our own way. This combination of our mother's DNA and our father's DNA is then passed to every cell in our body as this zygote divides over and over again to make the trillions of cells found inside the human body. And every one of these cells should have pretty much exactly the same DNA inside. Later in the year, we'll learn a lot more about how this combination of our mother's DNA and father's DNA gives us our traits, and how certain molecules inside our cells are able to read and interpret this universal genetic code. But for now, hopefully you understand a little bit more about this really important organic molecule that we call DNA, and how it is able to store and transmit genetic information from one organism to the next. Thanks for watching, and remember, you can always go back and watch this video again if you need a refresher on the structure and function of nucleic acids.